basically, and then going to do an internship at Streck Labs. Streck Labs does a lot of standards, so you got to be very precise in what you're doing down there, so that's going to help them along the way too. But I'd say one of the things for sure about Brett that I learned is he's very deliberate, which is a very good quality to have to go to graduate school. He would come to my office once in a while and then disappear for weeks, and I thought, is he doing anything? And then the next time I saw him, he had all this work done. So he does a lot on his own, and that's going to suit him very, very well in graduate school, which he's applying for. Graduate programs love students who leave the professor alone and troubleshoot on their own because they're very busy. And so it also tells you that he's got a lot of confidence in the way he thinks and what he does. So the results he gets are good. Also, my pleasure to then award him his honors. What do you even call them? Cords. So Dr. Bonner often does this, but he was nice enough to do that. So congratulations.
Awesome. Thank you again, Dr. Christensen. Again, my name is Brett Hilbers, and I'm a biology, chemistry, and psychology major here at Wayne State College. I really appreciate you guys coming and listening to speak about the research that I've done over this past year pertaining to ticks and the diseases that they carry. The way I'm anticipating this going is I'll speak for a while and then I'll open it up at the end if anybody has any questions. But just for an overview of the information that I will be covering in this presentation, will first pertain to ticks and their life cycles. We will be looking at the morphological features that they possess that allow them to survive. After that, we'll be looking at the pathogens that they are able to carry and the diseases that the pathogens are able to cause. After that, we'll look at a past study that was conducted a couple of years ago and how that led to some of the inspiration behind my study. And then finally, we'll cover the methods, results, and implications of my study. So to begin, what are ticks? Ticks are parasitic arachnids that are present worldwide, especially in humid areas, but they're present all over the United States. Their life cycle is going to be characterized by several stages in which they need to feed on a host in order to get blood to move to that next cycle. And the ticks are often going to benefit from the morphological features that they possess that make them very adept at feeding. And so to start off, the clawed legs that they have make them very good at positioning themselves. And then a unique set of mouth parts allow them to feed for very long amounts of time by first making incisions using the chelicerae that they possess, and then rooting themselves and anchoring themselves using their hypostome in order to feed over long periods of time. Because of the legs that they have with those claws, they are able to position themselves on grasses and other types of foliage with their arms extended, and when they sense hosts nearby, they are able to orient their bodies in such a way that allows those claws to attach, so that way they are able to begin that feeding process. They also possess palps, which are structures that allow them to sense a number of different sensory stimuli, such as carbon dioxide, heat, and vibrations, so that way they are further able to give themselves that opportunity to feed. And then finally, using the secretions that they are able to generate, they are able to anesthetize that site of incision, so that way the host is unaware that the tick is feeding, just to maximize that amount of time that the tick has in order to feed. You can really see how adept these ticks are at feeding just using these pictures, because you can see from the first day that they are unfed to that last day, they quite literally multiply in how large they physically are just based on how much they are able to feed. The tick's lifespan, as I mentioned earlier, is characterized by several stages, each of which is going to be based on feeding. And so in the early stages, after they hatch out of their eggs, they are larvae, and they will choose their first host kind of on a size basis, where they'll typically choose either a bird or some kind of small mammal at first. And this is really that first opportunity that ticks have to have those pathogens transmitted to them in that first feeding cycle, which after this point, they are able to pass to other animal reservoirs or humans, and we see this same kind of process happening through the later life stages of nymph as well as adult. We usually see these ticks most active during the early to late summer, and in these later stages they are typically going to prefer larger mammal hosts, and so those are often deer, humans, or really any other organisms that they are able to feed off of. At the end of their lifespan as adults, they copulate, and the females will attempt to have one more opportunity to feed, so that way they're able to get enough blood to nourish all of the eggs that they hold, and for a single tick this is typically going to mean up to about 8,000 or 9,000 eggs. In Nebraska, we do specifically see a couple particular species of tick, starting with the black-legged tick and the American dog tick, which are most pertinent to my research, and then also the lone star tick. Each of these types of ticks is typically going to carry a specific type of bacterial pathogen. For the American dog tick, this is going to be Rickettsia or Rickettsia, which is the causative agent for Rocky Mountain spotted fever, and for the black-legged tick, you're often going to see Borrelia burgdorferi, which is the causative agent of Lyme disease. Now, Lyme disease and Rocky Mountain spotted fever, in their initial symptoms, are often going to resemble one another, because they're often going to be characterized by rashes as well as just low-grade fevers. But if gone untreated, severe cases can often lead to complications such as heart arrhythmia, heart palpitations, and also central nervous system damage. And so, when gone untreated, these diseases are certainly dangerous to really individuals of all ages, just due to the fact that the ticks are able to infect all humans. In light of this, a previous study was done a couple of years ago by researchers at the University of Kearney. Dr. Christensen and I had the opportunity to listen to one of these researchers, Dr. Schaefer, who had presented to two of us and many others in the life sciences department about the research that they had done using ticks gathered from south central Nebraska. And so they had gone along the Platte River, and they had gathered a number of ticks, and had similarly in my study had done DNA extractions, which they had analyzed, and they had found a fairly significant presence of rickettsial bacteria, which again are those bacteria that could cause Rocky Mountain spotted fever. And after this, this kind of led to the idea that led to our study, which was conducted in Northeastern Nebraska instead of South Central Nebraska. And so we were testing upon the hypothesis that we would be able to find evidence of pathogenic bacteria and viruses in Northeastern Nebraska by gathering ticks from the Wayne and Stanton counties, and then performing bacterial isolations, and then finding DNA extractions and analyzing that DNA. So to start with the bacterial isolation of the process, every single tick that we had gathered, we would cut in half physically with a scalpel, and half of that body would be put into a test tube full of liquid media. Now, media is basically just a term that we use to refer to bacterial food, and so by letting that sliced half of that tick stay in that liquid media overnight, we were able to let all those microorganisms living inside that tick proliferate, so that way that next day we could take an inoculating loop, which is just a little metal loop, to go inside that media, pull out a bunch of those bacteria that were able to grow overnight, and we were able to make a street plate out of them. And the benefit of street plates is that we are able to isolate specific colonies. And so on this right half of this dish, you can see fairly dense growth in the bacteria that grew, and we can't feel confident that there would be any isolated specific species of bacteria right there. And so instead we allow that inoculating loop to deposit a lot of that bacteria, so that way when we get to this other side of the dish, we have these small little dots of colonies that we can take. We can put those into a new test tube to have that isolated colony of bacteria proliferate inside the liquid media, which we would then bring back to one more streak plate to use in our biolog bacterial identification test. Now, these testing plates are going to work off the mechanism that different types of bacteria are able to digest and interact with different types of metabolites. And so by putting a different metabolite in each of these 96 wells and putting a little bit of the bacterial colony that we've isolated into these wells, we are able to see whether or not that bacteria can digest that particular metabolite based on whether or not a dye is present and is visible for us. And so we can input all of the results of each of these wells into the database that Biolog provides, and they will give us the results of whether or not the bacteria that we isolated is one that they have in their database as medically relevant. And so there were a number of different bacteria that we did end up isolating, but I'm just going to share a couple that were a bit more medically relevant than others, starting with Pseudomonas butida. This was the only bacterial colony that we had isolated that had a history of causing disease in human patients, and this was often seen in immunocompromised individuals in hospitals. And we also see Cyanobacillus xylem exitans and Pseudomonas congelans, which, if anything, actually displayed antimicrobial properties. It was interesting because we had set out looking for bacteria that caused for a disease in humans, but we had actually found some bacteria that, if anything, actually acted against fungal microbes, and so that was an interesting finding. And then if we go back, remember there's still that remaining half of that tick that we had sliced, and so that other half of the tick went towards this DNA extraction portion of the study, and so we had placed that other half of that tick inside a test tube, and we had used it in a bead beater procedure. And so by loading up that test tube with a bunch of microscopic glass balls and then putting them into that bead beater, that is that top image there, the bead beater would shake that tube so much to the point where those microscopic glass beads could break all of those cells of the tick and then of any microorganisms living within inside that tick, and that would give us a resulting lysate, which is basically just all of the intracellular fluid and all of those cellular constituents from all of those microorganisms that we had within that sample. And we would use that remaining lysate in the remaining part of the procedure, where we would first use a Promega DNA isolation kit, which helped us to isolate that DNA from the rest of those cellular constituents that were present within that sample. And so by adding different solutions within the kit and then centrifuging them, we were able to sequentially remove different constituents of that sample to more or less get an isolated sample of that DNA that we were looking for. And so what centrifugation is going to do is it's going to spin our samples around and to generate so much force on those samples that we're basically sedimenting and separating all of those cell constituents based on how dense they are. And so by doing that, by adding those solutions and centrifuging them, we're basically removing one thing at a time until we have a more or less purified sample of DNA that goes on to the PCR segments of the procedure. Now, PCR, if summed up, is basically the replication of targeted sequences of DNA that we have in our sample. And so, if we're looking at this process here, what we are going to start with are DNA polymerase into nucleotides, or DNTPs. These are basically the building blocks of DNA, as well as the cellular machinery that we need in order to produce DNA. And we are going to add our template strand, in other words, just that DNA that we had isolated from our sample. And the DNA primers is really where we start that specificity and targeting, because we were looking for those aforementioned bacteria that we had introduced earlier in the presentation. And so by designing primers that have specific sequences that line up with the sequences of those microorganisms that we are looking for, we can selectively target for those specific types of microorganisms. And so by taking our double-stranded, double-helix DNA and subjecting it to high amounts of heat, we are able to separate those double strands just long enough for our primers to bind if in our sample there's a corresponding sequence. And by heating our DNA, or by heating our sample back up to a temperature in which our polymerase, that is the machinery that we need in order to produce the DNA, by heating it back up, if there were any primers that did bind due to that correspondence of the primers that we designed in the sample that we obtained, that polymerase can make new DNA strands. And what's cool about PCR is the fact that it isn't just those original DNA strands that are used to produce all of the DNA that we have as a product from this procedure. Every single subsequent set of DNA that we make is used as a template for that next cycle. And so after 30 cycles, even if we've only started with a very little amount of DNA, after 30 cycles, which only takes about 2 hours, we have a little under 1.1 million copies of this specific strand of DNA that we have targeted. And so we took those PCR products and we loaded them up into a gel electrophoresis test, which is going to take advantage of the negative charge that our DNA possesses. And so by loading our DNA into these little wells here, positioning them next to the negatively charged electrode, and then having a positively charged electrode on the other end of our apparatus, we are able to provide power to that entire apparatus here, and due to the fact that opposites are going to attract, that negatively charged DNA is going to be repelled away from the negatively charged electrode towards the positively charged electrode. And due to the pores that are physically integrated within the gel that we made, the smaller the DNA band present, the faster it's going to move through. And therefore, if we have it running over a set amount of time, those smaller bands are going to move faster and further than those longer bands, which are going to move slower. And so it's horribly difficult to see. But essentially, every single one of these bands that we see here is a primer dimer that's formed by negative test results. Okay, that's slightly better. So you can see all of these primer dimers that form that bottom layer, and all of those show all of the negative tests that we had. It's basically those primers that we had added in that first step of PCR, right? Those were the smallest little segments, and so those are what we see showing up. But in this lane, we see the absence of a primer dimer, which is interesting, but then we can also see a band forming right below that arrow there. And that there is evidence of an organism within that sample having corresponding DNA to the primer that we designed. And in other words, we would have evidence of Rickettsia bacteria living within that tick that we had sampled. And so we ended up having 2 positive test results that both used that same primer, which gives us 2 positive results for the Rickettsia bacteria that we had introduced earlier. That's that bacteria that can cause Rocky Mountain spotted fever. And both of those ticks that carried that DNA were obtained from a lake near Hilbers, Nebraska. This ultimately represented 2 of 70 ticks that we had tested, a little under 3% of the total ticks that we had tested, and a little under 10% for the ticks that we had tested from this area. So in regard to the implications that result from this study, we saw how dangerous Rocky Mountain spotted fever and Lyme disease can be in untreated cases. And so given evidence from this study, as well as the study that I mentioned earlier from Lutke, it's very important that healthcare providers recognize the signs and symptoms and are aware of treatments for Lyme disease, Rocky Mountain spotted fever, and other tick-borne diseases, especially if they're becoming more common. And studies such as these are only going to become more important, not just for Lyme disease, but for any disease in which animals can be a reservoir and in which we see other organisms serving as vectors. And so, for example, coronavirus is a virus that we typically see harbored within animal reservoirs. And so studies such as these can help detect and make the general population aware of the risks that exist in a given area for a lot of these vector diseases. And then finally, one more implication that I wanted to list was just the fact that in both my study, which I've gathered the ticks that have tested positive from around a lake region, and the blood key study in which we see the ticks gathered from around the Platte River, I think it might be interesting to look further into the role of waterways and how these natural bodies of water form habitats that the animal reservoirs that animal vectors are able to pick up disease from. I think it'd be interesting to further look into the role that these water habitats hold for the process of disease transmission. So I would like to take a moment now to acknowledge and thank my advisor, Dr. Douglas Christensen, and really all of the life sciences and physical sciences and mathematics departments at Wayne State College. It's because of them that I've been able to research and develop such a love for doing research and hopefully follow in their footsteps and emulate them one day. So, at this point, if there are any questions for me, I'd be happy to answer them.
Just curious, what was the species breakdown for the ticks that you found in Wayne and Stanford counties?
In the bacterial part or the
All of them.
DNA structure, all of them. So a lot of the bacteria that we found just in the live bacterial isolation part of the procedure were...
Oh, sorry, the tick species.
Oh, the tick species, yes, sorry about that. So every single tick that we had out of those 70, they were all American dog ticks.
Okay.
We were thinking we might find Lone Star ticks or black-legged ticks, but we had not. And we had taken ticks from the Prairie Reserve, some in Pilgrim, and there were just some surrounding areas outside of that, some like creeks around Wayne. So
What time of year was it?
we gathered these during the summer, is that right? Yep.
So most of them are summer.
How long stars? Was it just grassland or wooded areas?
I suppose a mix, because the Prairie Reserve that we have is, it's a lot of grassland, but there are a number of trees that kind of surround it, and I can't speak to the other areas because we did not physically go out, or at least I did not. I'm not sure if Dr. Christensen physically went out and gathered these, but...
Pilgrim is also just grassland.
Okay.
Surrounding trees.
It makes a little more sense than not something monsters.
It was just a summer, just dragged the clock back around. We took over once we had frozen ticks.
Okay.
Can you go back to like the symptoms?
Yep.
Photos, and what are those actually this caused from?
So, for each of these, this leftmost picture is Lyme disease, and then the picture on the right is Rocky Mountain spotted fever. And then similarly, you see Lyme disease on the bottom, Rocky Mountain spotted fever on the top. That inflammation is going to be due to damage to blood vessels, and so at first, just the site around the site of that incision and feeding shows up, but we do eventually see, in those untreated cases, we see that inflammation spreading throughout the body in both cases, but they do have slightly different looking rashes, and so, you know, you see kind of, I guess, just homogeneous looking spreads of inflammation on the skin for that Rocky Mountain spotted fever case, and then you kind of see bigger lumps for Lyme disease.
So with, since I'm not clinician, do they treat this with doing inflammation? Is it anti-inflammatories or is there other drugs that they use?
Yes, primarily anti-inflammatories, and it's not something that I extensively covered, but a lot of times due to how common, you know, rashes are to just diseases in general, a lot of times these tick-borne diseases are misdiagnosed and mistreated, and so for that reason, you know, it's a bit rare with Lyme disease just because, like, with that bullseye pattern, like it's a bit more I suppose it's a special type of, it's a special recognized pattern that clinicians are able to recognize. And so, especially in the case of Rocky Mountain spotted fever, in which that rash does resemble the rashes of a lot of other diseases, anti-inflammatories would be used, but there would also be that risk of, if you're only treating for the inflammation, you might miss the other signs and symptoms that can, be spreading throughout the central nervous system as well as the cardiovascular systems, and so there are other drugs that are needed to counteract those symptoms as well as the anti-inflammatory drugs. Yep.
What drew you to this research to begin with?
Well, I remember when Dr. Christensen and I had listened to Dr. Schaefer, right? She was one of those individuals who had presented that initial research that had been published in 2019. I think that was really the first opportunity that I had to listen to and understand academic research, that wasn't super popular. And so the idea of going out into the field and collecting evidence of, underlying phenomenon, I think that idea really captured me. And so being able to kind of contribute to this type of field and contribute to our understanding of tick vector diseases in this way, it's something that really scratches my itch for being someone who discovers and researches things that matter to people, things that affect people in a very real way. And so ultimately I think that experience for me is something that really drew me into this research and really developed that level of research that I have at this point.
So you're applying to graduate school, in what area do you migrate to, hoping to be here?
So every single program that I've applied to has the same kind of 1 year serving in a rotation of labs in which you're kind of just doing everything, and then after that you would be placed into a specific lab based on your own interests. And so at least what I had indicated on all of my statements was that I would be interested in going into studying microorganisms, either from a microbiological or biochemical standpoint. And so either studying microorganisms similarly to how I did in this study, or studying proteomics. I think that proteins and enzymes are just fascinating, and so using bacteria as kind of a case study for how we can study and maybe even engineer proteins. I think all of that work is just delightful because it represents a lot of sustainable solutions for problems that we have that we could counteract through enzymatic means. So I'm hoping to kind of do something similar to this, but perhaps in more of a clinical setting than going out into the field and doing ecological studies.
So you're not applying to graduate school, so you don't want place?
No, but wouldn't that be nice if I did? All right. Well, thank you all for being here again. I know they're presenting other projects throughout the day, so if you're interested in continuing to learn about stuff, I know that we have presenters in this room. So, yeah. Thank you all.